Well, hello. Welcome to another Tink Talks lecture. Today's lecture will be geared for a high school biology class, and this will be a lecture on microbiology and just a general overview of the topic of microbiology. So, nothing in depth, everything very superficial and on the surface. This is a lecture that's designed for my ninth grade biology class that I teach, but of course, if you're taking microbiology or you're learning about it in your biology class, maybe this could be useful for you as well. So let's get started. This is part one of a three-part lecture on general introduction to microbiology. So welcome to the wonderful world of microbiology and infectious disease. A wonderful world that it is. So what are microbes? Microbes are living things that are too small to be seen with the unaided eye. So you see the prefix micro, which of course means small. And so that's what microbiology is all about. Small living things. And the main categories of microbes are bacteria, fungi, protozoa, and algae. Viruses can be put in there as well. However, sometimes viruses are not considered to be microbes because some scientists, microbiologists, consider them to be non-living things, but you're going to learn about them in any microbiology course. So we could say there are 5 categories, I suppose: bacteria, fungi, protozoa, algae, and viruses. Now, typically, we view microbes as harmful, always causing disease. But guess what? Only a minority of them is pathogenic, and pathogenic means disease producing. And it comes from 2 words, the prefix pathos, meaning disease or something that's going to be bad occurring to a living thing, and genic, which means the beginning of. So the beginning of disease or disease causing. And the reason why we think that is because we pay so much attention to those microbes that cause disease. We're concerned about them. They make us sick, and we're concerned about our lives, obviously, and we don't want to get sick. And so many of the microbes that we pay attention to are those that we would consider, quote, bad, the ones that are going to make us ill. But that's a very small minority of them. And in all of those categories, those 5 categories of microbes, bacteria, fungi, protozoa, algae, and viruses, all of them have pathogenic members, but most of them are completely non-pathogenic. In fact, they're extremely helpful, especially in terms of ecology and ecological microbiology. There's so many connections between the health of an ecosystem and the microbes that are living within that ecosystem. Lots of microbes, a varied number of microbes, high diversity of microbes in an ecosystem, better health. Fewer microbes, less diversity, usually leads to a lesser degree of health within an ecosystem. That's generally speaking, but that's often the case. And that's even the case within us. I mean, we think of ecosystems being outside, but we have ecosystems within us. They're micro-ecosystems, but nonetheless, they're ecosystems. And so the greater variety that we have within us, that means that we have typically better health. Not always, but again, I'm speaking generally. So we typically only pay attention to those that are the bad guys, when we should also pay an equal amount of attention to those that are beneficial, which we do, but we typically don't talk about them as much. So what do they look like? So here's just a representation of a few different ones. On the upper left, we have a micrograph of bacteria. On the upper right, those would be fungal spores, which creates some problems for people when they inhale them. On the bottom left, that is a species of algae. And then on the bottom right, there are protozoa as well. So this is just a representation of the categories of the microbes and viruses as well, which is part of this lecture, which will be part 3, where we talk about viruses. So let's start with bacteria. Bacteria are microbes, single-celled organisms, too small to be seen without a microscope. They are never multicellular. However, they can grow in masses that kind of take on a multicellularity about them, but that's later. We'll talk about those later. That's in what's called a biofilm. They're considered to be prokaryotes, which means that their DNA is not enclosed by a membrane. And we talked about this in my biology class earlier this year when I saw all of your smiling faces before this pandemic that we are currently in. And we talked about the two main categories of cells, the prokaryotes and the eukaryotes. Prokaryotes comes from the Greek terms pre-kernel, but kernel as in like a seed for karyote, but that doesn't make a whole lot of sense. So let's say pre-nucleus. And so these were organisms that appeared on the scene before nuclei were incorporated into cells. Eukaryotic cells, they have a membrane-bound nucleus, and EU means true. And so these are true nucleus cells. They have a true nucleus. So where can bacteria be found? Well, pretty much everywhere. So there's a term that we use for in reference to the presence of bacteria and where they can be found, and that term is ubiquitous. Ubiquitous means everywhere, and so often you talk about the ubiquity of bacteria. You'll see it in micro textbooks, and you'll talk about it if you have a microbiology course. But they're found everywhere, and to say that they're ubiquitous is just a fancy way of saying they're everywhere. They're within us, they're on us, they're in the air, they're in the environment, they're on your pet cat who happens to be sleeping on your couch right now. And there are billions upon billions of them within your very soundly sleeping cat. Same with us. We've got trillions of them on us and within us. Here's what bacteria typically look like. The picture on the right is a micrograph from a transmission electron microscope, and the one on the left, of course, is just some computer-generated picture of one. And you can see the main structures that are found within bacterial cells. We'll be talking about some of them. You'll notice that in the middle of the cell, on the one on the left, it's clearer to see, there's this tangled mass of nuclear material, and that's called the nucleoid. That's where the DNA is. Now, that is not a chromosome. A chromosome, by definition, is something that is wound up around these proteins that are called histones and have a very distinct and organized structure to them. That's not the case with the bacterial DNA. It's just a long strand of it. And there's also a circular piece of DNA, which you can see labeled in the picture. They're called a plasmid. And a plasmid is a separate circular piece of bacterial DNA. It's often used for genetic engineering experiments and applications. There are no organelles within a bacterial cell. The closest thing the bacteria have to organelles would be, I suppose, ribosomes. Those are cellular structures made of RNA and proteins, but they have no membrane around them. And so by the definition of an organelle, I suppose they can't really be considered an organelle. However, they carry out a very, very important function that is required for all cells, and that is the process of translating RNA into proteins. There's also other structures in there that are kind of similar to organelles, and you'll see labeled on the left there called an inclusion. And inclusions are just these structures that are included with a bacterial cell, and they perform a number of functions that help with their metabolism or storing things. But they're not an organelle. They're mainly storage facilities within the cell. They have a cell wall that is extremely important for them and enabling them to survive. And then on the outside of them, they form this capsule, and there are a couple of different ways that they form this capsule as well, which is important to their survival in adverse conditions. They have these root-like structures that are sticking out of them. See them? They look like crazy, wacky hairs sticking out of the surface of them. They're called fimbriae. And think of fimbriae they're cell membrane extensions that enable them to adhere to, say, a mucous membrane or to sometimes each other as well. They're kind of like roots, I guess you could say, that enable them to root themselves within a soft tissue or membrane. And then they have flagella. The flagella are pretty advanced. They have pretty sophisticated flagella, and they need that, to enable them to propel themselves through fluids, whether it be water or sewage or your colon, your large intestine, which is where E. coli reside. So the flagella are necessary in order to propel them through these fluids. So let's look at some of their parts. The capsule, it's the outer covering. Some bacteria protects a cell and makes them more pathogenic. Why? Well, that capsule can make it almost near impossible for a white blood cell to break down the bacterial cell. It's considered a glycocalyx, or what you could say a sugar coat. It also is called a slime layer as well. Depends on the organization of it. If it's highly organized, it's a capsule. If not, it's called a glycocalyx or a slime layer, not as tightly packed and organized. So this capsule enables them to survive in dry conditions, adverse conditions. It makes them able to survive these conditions so that they don't become desiccated or dried out. Enables them to survive in what would normally be hypertonic conditions for them. So it's important for their survival and also for their success as well. They have a cell wall. It's a semi-rigid structure. Protects the interior of the cell from changes on the outside, and it's made of this protein-carbohydrate complex called peptidoglycan. Peptidoglycan is found nowhere else in the biological world. It's only found in bacteria. Some bacteria have just a few layers of peptidoglycan, while others have many layers of them, and it's a very repetitive type of structure where it's got basically these two carbohydrates that repeat each other, and then they're kind of all held together by ropes made of protein. So think of it like a log raft, where the logs would be the proteins, which are shortened to, the names of them shortened to NAG and NAM, which stand for big words that I'm not going to get into now or tell you. But it's basically just these repeated carbohydrates that form kind of these log-like structures, and then these protein ropes that hold them together. And so we call it peptidoglycan. If you look at the words, you can see peptidoglycan refers to both of those. Peptid refers to proteins, and glycan refers to sugars or carbohydrates. And so that basically tells you what its structure is. And like I said, it's unique. Bacteria have it. Nobody else does. And they feel pretty special about that, I'm sure. They have a plasma membrane, which is a non-negotiable for all living things. There has to be a plasma membrane for everything, from bacteria to humans and everything in between. There needs to be a plasma membrane. And it's a phospholipid bilayer, as it is in all cells, and it's got embedded proteins in it and carbohydrates as well. Materials can enter and exit, so there's a lot of cell transport that occurs through the plasma membrane as well. It's quite similar to eukaryotic cell membranes as well. Eukaryotic cell membranes have a few more bells and whistles and are capable of carrying out a few more varied processes, but the basic structure is the same. Ribosomes, they appear as small dots in the cytoplasm. They're required for making proteins through the process of translation, which bacteria must carry out, as well as all cells must. And the nucleoid is the area where the DNA is found in the cell. And if a term, by the way, ends in OID, that means it's like its prefix. So a nucleoid is like a nucleus, but it's not a nucleus. The DNA strand is a long, continuous, usually circular, but not always, wound up mass of nucleic acids. And bacteria possess no chromosomes. So we don't want to say it's a singular chromosome, which I've seen in some articles and so forth. And you can't really say that, because by definition it's not a chromosome, because in order to have a chromosome you have to have the DNA wound up around these proteins that are called histones, and bacteria do not have histones. It's a disorganized mass of DNA. They have, like I said, these plasmids, these separate small circular strands of DNA. They're not connected to the main DNA strand, and we have taken advantage of these plasmids and utilized them in genetic engineering and incorporating these bacterial plasmids into eukaryotic cells so that we can get the production of desired proteins, for example, like insulin, which was discovered we could use bacteria to our advantage, using plasmids from E. coli in order to create large amounts of insulin for those who need it, who are diabetic. Cytoplasm, everything inside the plasma membrane, mostly made up of water. It's the same in eukaryotic cells as well. We say the cytoplasm is everything in between the nuclear membrane and the cell membrane in eukaryotic cells, which is basically going to be the same substance in prokaryotic cells as eukaryotic cells. Pili. These are hair-like structures that allow bacteria to attach to other cells and transfer DNA. And as I mentioned in the picture of the cells, remember those root-like structures called fimbriae? Well, they have hundreds of those, sometimes thousands of those, fimbriae that is, per cell. Well, pili, they only have a handful of them, like less than 10. They use these pili mainly for transferring DNA through a process called conjugation. And the way this works is that the pili will attach to other cells, and through the pili they will transfer DNA, and this strengthens neighboring bacteria. It leads to the passage of mutations, some of which can be beneficial. Pass those mutations on to other cells. Mutations such as resistance to an antibiotic, for example. And through this transfer of DNA, some bacteria, those that have developed resistance against a particular antibiotic, can then transfer this resistance to other bacteria through these structures called pili. So they enable the bacteria to basically, it's not a form of reproduction. They're transferring DNA. Don't confuse it with that. That is not the way they reproduce. They reproduce asexually. They do not exchange DNA. They reproduce mainly through a process called binary fission, which is an asexual form of reproduction. They just make clones of themselves. But here we have transferring of information, not for reproduction, but for strengthening of cells and cell-to-cell communication. And then this leads to the production of, like I mentioned earlier, biofilms, which we'll talk about later. And flagella, these are these structures mainly made of proteins, microtubules. My bio students should remember those structures, microtubules. Hollow tubes of proteins that enable the bacteria to move through the medium that they happen to be living in. Shapes of bacteria. They come in a variety of shapes, but these are by far the three main shapes. You have bacillus, which is picture number 1, coccus, which is picture number 2, and spirillum, which is picture number 3. And of those three, bacillus and coccus are the most common. There are a number of spirilla bacteria, but bacilli and cocci bacteria are the most abundant. That's their most abundant shape. Why? It's their favorite shape. I don't know. It's most convenient for them, for their ability to live in the different environments that they are in. So these are the three main shapes. There are probably, I don't know, around 7 or 8 other different shapes that they take. Some of them have no real distinct shape at all. They like to change shapes. They're goofy that way, schizophrenic, I don't know. But they're few and far between. These are the three main ones. So how do they arrange themselves? Well, they come in these different, how do they join cell to cell? Are they rogue? Are they living on their own? Do they like to live in groups or what? So they are basically three arrangements. Straight chains of bacteria referred to as strepto. Strepto means chain-like, linked together like streptococci, the one that causes, say, cavities on your teeth, Streptococcus mutans, one of the primary culprits of dental decay, or Streptococcus pyogenes, which causes strep throat. So they're chains. They exist as pairs, which is called diplo, or they exist in clusters called staphylo. So I mentioned Streptococcus. So Streptococcus, like you see in the picture there, see those the chain of purple circles there, strepto. That would be Streptococcus because coccus are round cells, or Staphylococcus. That would be a cluster of round cells. Or Diplococcus, for example. That would be a pair of round cells. So we take those terms on the previous slide and we combine them with these, and then we give the names to bacteria. Now, not all bacteria are named in this way, but a number of them are. So we use characteristics of the bacteria in order to name them, like Staphylococcus aureus, for example. Staphylo, clusters, coccus, round, aureus means golden. And if you're thinking of the periodic table of the elements, which I'm sure you were, what is the chemical symbol for gold? Au. Yes, and so Staphylococcus aureus, A-U-R-E-U-S, aureus. So golden colored bacteria. Gram-positive and gram-negative. So these are the two broad categories of bacteria. If you could divide them up into two basic categories in terms of, at least in terms of their cell wall structure, this would be it. This was developed in the 19th century by a scientist. His last name was Graham. And he figured out that staining the bacteria, he could kind of differentiate them into two broad categories, gram-positive and gram-negative. And it's, like I said, based upon the structure of their cell wall. The gram-positive bacteria have a really thick layer of peptidoglycan. And then they have their cell membrane. Gram-negative bacteria have an outer membrane, then they have this thin layer of peptidoglycan, and then they have their inner cell membrane or the plasma membrane. Now, we found out some interesting things about this. It's more than just their structure. We found out that gram-positives are generally easier to treat and kill due to the simple composition of their outer peptidoglycan layer. Gram-negatives tend to be more difficult to remove, get rid of, neutralize, kill because of the complex nature of their outer membrane. And so it's more than just, well, we got this type of cell wall and we have this type of cell wall. We found out more things through experimentation and discoveries that it means more than just a simple, we could say, anatomical classification. So what makes it gram-positive easier to treat or kill? Well, the peptidoglycan, like I said, is just proteins and carbohydrates. If you can tear apart and take down that peptidoglycan, that wall of protection is gone and it's pretty easy to get rid of. Remember I said the peptidoglycan is like a log raft held together by protein ropes? Well, if you take the ropes and you cut the ropes of a log raft, what happens to the raft? Well, if it's not held together by glue, the raft is going to fall apart. And that's what happens to the gram-positive cell wall, the peptidoglycan. So we have developed antibiotics and antimicrobials and disinfectants that are really good at tearing apart the peptidoglycan, basically snipping through those protein ropes. And once you do that, it's over for the bacterial cell. They're going to die. They're going to spill their guts out. Gram-negatives, though, they have this outer membrane it has a lot of bells and whistles with it. It's got a pretty complex nature to it. And so as a result, it's difficult to break down that wall, that barrier, compared to the gram-positive simple peptidoglycan. So yeah, it's a thinner cell wall and cell membrane with gram-negatives, but it's the complexity of the gram-negative that makes them, in some cases, more pathogenic and harder to kill. What do they look like? Well, you've got the gram-positive on the left, and I'm positive that's gram-positive. So you'll notice it's got a thick layer of peptidoglycan, and then it's got the cell membrane inside. As compared to the gram-negative on the right, you've got the outer membrane, which is complex, and this picture is not doing the complexity justice. There's more to it than what you see in this picture. And then it's got a thin layer of peptidoglycan in the middle, sandwiched in between the membranes, and then inside is the plasma membrane that they both share in common. And like I said, you get rid of those protein ropes that hold it together, and you break down, and you can see in the picture there this fancy acid called teichoic acid. If that's broken down, that's what leads to the destruction of the cell too. It adds strength to the peptidoglycan, and so you remove that strength and it falls apart. So what are some examples? Gram-positive bacteria, Staph aureus is gram-positive. The streptos are gram-positive, like Streptococcus mutans, like I said earlier, causes cavities and dental decay. Gram-negatives, those would be E. coli as a gram-negative. Another common one that I'm pretty sure you haven't heard of, but it's common in the bacterial world. It's called Serratia marcescens, and it's a gram-negative. And so we can take these bacteria, we stain them through this gram staining mechanism, and you can look at them under the microscope. And if they turn out red, they're gram negative after the gram staining procedure that you carry out. If they turn out purple under the microscope, they're gram positive. And so there's a series of steps that you follow called the gram stain. And at the end of it, you then look at the bacteria under the microscope on a slide. And you'll see that the cells will either be red, indicating gram negative, or the cells will be purple, indicating gram positive. And so it's a very easy procedure to do as long as you have the correct chemicals to stain the cells. It's very simple, and all you need is a light microscope to do it. This is, like I said, what they look like. Gram positives are purple in color, and gram negatives are red in color. And that's exactly what they look like under a light microscope. So, talking about normal flora or microbiota. The old term was flora. It's still used today. The reason why we called it that was when we first discovered bacteria, we weren't sure what are we going to classify these organisms into. Are they plants? Are they animals? I mean, back when they were first discovered in the by Leeuwenhoek in the late 1600s and early 1700s, of course, it was a brand new form of life that we had seen. He called them animalcules, and then as we started noticing them and doing more observations of them, people were scratching their heads. Are they animals? Well, no, they're not animals, so let's just say they're plants, and that's how they were classified. So we called them flora. The more, the more learned term, I guess we could say, is microbiota. So this term, normal flora or normal microbiota, it refers to the microbes that are living on us and in us. And we have a huge variety of them. And these microbes make up, the ones that make them up are fungi, protists, but mostly bacteria are living on us and in us. We have about 10 to the 13th cells. That's a lot of cells. We're in the trillions there, of course, and we harbor about 10 times as many bacterial cells than human cells, about 10 to the 14th bacterial cells on us and in us. So we have an enormous number of bacteria that are influencing us in a number of ways, physiologically speaking. Now, psychologically, I don't know, people get into that. They talk about how bacteria affect us psychologically. Not sure if I buy into that. I guess I have to do a little more research. But definitely, most definitely, physiologically, they are affecting us. Certainly they are. I mean, how do we digest our food? Bacteria. Extracting the nutrients from them and so forth. In most cases, these microbes are beneficial to us because they protect our body from diseases by preventing the overgrowth of harmful microbes. That's called microbial antagonism. So we have this staph species, for example, living on our skin, especially referring above our waist. About 90% of all the bacteria, all the microbes even, that are living on our skin from the waist up is this lowly staph bacterium called Staphylococcus epidermidis. And you may recognize the term epidermidis from the word epidermis, which is the covering of our skin. 90%. And so Staph epidermidis is normally a completely non-pathogenic bacterium. And as a result of having so many of them living on us, they are basically taking up residence on us and protecting their turf and not allowing pathogenic microbes from getting on our skin and creating some kind of skin infection or infestation or something. And so as a result of all of these Staph epidermidis cells, we've got this bacterium that is protecting its turf and not allowing other bacteria to take up residence. And again, that's called microbial antagonism. So it's not a direct benefit. We're not directly benefiting from Staph epidermidis. I mean, they're not giving us something in return that we need. That would be mutualism. It's more along the lines of what's called commensalism, in which they benefit, one species benefits, that would be the bacteria. They benefit and we are neither helped nor harmed. We are helped kind of indirectly, but not directly by these microbes. Where can we find them? Where can we find these microbes on us and in us? They can be found throughout the body, localized in certain regions. I mentioned the skin. We're covered in bacteria and loads of different microbes. Fungi reside on our skin as well, and we have literally billions upon billions of them. And we are prime time real estate. I mean, this is where the microbes want to be. And they want to take up residence there. So the next time you feel kind of down and out, a little depressed and thinking nobody likes me, everybody hates me, guess I'll go eat worms. Remember, you've got bacteria that love you. And they think you are the best. So they're all over us, all over our skin. They're on the surface of our eyes on a membrane called the conjunctiva. And there's ways that we can protect ourselves from getting eye infections from all these bacteria that are thriving on our conjunctivas. Nose and throat and the upper respiratory system. In our mouths, there's loads of bacteria in there. I mean, our mouths are open. We're talking a lot. We're breathing, and bacteria just naturally will go in there, of course, in the nasopharynx region and in the mouth as well. The large intestine is I was going to say polluted with bacteria and microbes, and I shouldn't because pollution seems bad, but it's good. We are polluted in a good way with all these microbes that are living within our large intestine. I mean, it is a very diverse ecosystem that is going on in the large intestine. In our urinary tracts as well, there are some bacteria, and in the reproductive system too. We can find all these. These are all regions where normal microbiota can be found. Some places many more than in other places, but nevertheless that's where we find them all the time. They're continual residents of those regions of the body. So it's nice to know that you've got friends to accompany you on this journey of life, and we call them the normal flora. Well, that's the end of part one. I'm going to stop here. I don't want to go on too long, and I'll pick up with part two of this introductory lecture on microbiology. And thank you for watching.